Each cell contains a complete copy of the organism's genome. A gene that is transcribed is said to be expressed. Not all cells express the same genes, which is why different cells perform different functions. Even within the same cell, different genes will be expressed at different times and perhaps at different levels. A few exceptions are housekeeping genes, which are in constant use to maintain basic cellular functions. Differential gene expression, that is, when, where, and in what quantity, is what makes cells different. There are many applications of microarrays and related technologies. In cancer research, we may be interested in the molecular characterization of tumors on a genomic scale to provide a more reliable diagnosis and identify more effective treatments for cancer. In immunology, we can study the host genomic responses to bacterial infections. Many scientists perform studies in model organisms such as mice, rats, and zebrafish. These are multifactorial experiments designed to monitor expression response to different treatments and dose levels over time or in different cell types. We may be interested in comparing mRNA transcript levels in different types of cells to determine what genes vary by tissue type, treatment administered, state such as tumor versus normal cells, what is different between different strains of the same organism, and how gene expression changes over time. In this module, we have seen that our cells include DNA, and all cells in our body are lineal descendants of a single fertilized egg. Therefore, in normal, healthy humans, almost all cells carry genomes that are reasonable, accurate copies of the genome that was initially present in the fertilized egg. However, you have no doubt noticed that cells throughout your body are phenotypically distinct. For example, your skin cells are very different from your brain cells, even though they are genetically identical. This is due to differentiation, which is the process whereby cells in different parts of the embryo begin to assume distinct phenotypes. Differentiation can be understood by examining the set of genes that are expressed or transcribed in some cells but not others. That is, there are tissue-specific genes. These tissue-specific genes may be due in part to transcription factors. Transcription factors are proteins that bind to specific DNA sequences in the control region of each gene and determine whether or not the gene will be transcribed. The specific stretch of nucleotide sequence to which the transcription factors bind, often called a sequence motif, is usually quite short, typically 5 to 10 nucleotides in length. Some transcription factors provide RNA polymerase enzyme with access to the gene, while other transcription factors block such access to ensure the gene is transcriptionally repressed. Histone modifications may also affect transcription by RNA polymerases of specific regions of chromosomal DNA. Additionally, methylation of CpG sites and also microRNAs affect gene expression. We will discuss methylation later. Cancer is a disease in which cells escape the restraints on normal cell growth. In fact, cancer is not just one disease, but a generic term used to describe a group of more than 200 diseases sharing common characteristics. Namely, cancer cells are insensitive to inhibitory or anti-growth signals. They evade apoptosis, which is programmed cell death, so they seem to have limitless replication potential. Note that normal cells undergo around 60 to 70 replications, and cancer cells have the ability to form new blood vessels to continue being fed, a term called angiogenesis. To date, no single cancer-causing gene has been discovered. There are, however, some known mechanisms whereby mutations and genetic alterations cause cancer. This includes gain-of-function genes, called proto-oncogenes, loss-of-function genes, called tumor suppressor genes, and translocations, which give rise to novel protein products through aberrant gene expression. Cancer is an inheritable disease, at least within a person from cell to daughter cells, such that once a cell has become cancerous, all of its descendant cells are cancerous. Pathologists make an interpretation based on a compendium of knowledge which may include morphological appearance of the tumor, histochemistry, immunophenotyping, cytogenetic analysis, etc. This is an example of a slide a pathologist may use for making the diagnosis of diffuse large B-cell lymphoma. When using microarrays to profile thousands of genes simultaneously, researchers identified a signature that separated diffuse large B-cell lymphoma patients by clinical outcome, namely overall survival. When looking at traditional demographic and clinical characteristics that separate diffuse large B-cell lymphoma patients into high and low risk groups, using gene expression data, the low risk group could be further refined into good and bad outcomes. Such information may be helpful to clinicians to ensure they administer more aggressive therapies to patients unlikely to achieve long-term survival benefit. So, an application of high-throughput genomic technologies is improved cancer diagnosis, that is, to identify some classes by dividing morphologically similar tumors into different groups based on response. More reliable tumor classification and subclassification should in turn lead to the administration of more appropriate treatments, thus leading to improved outcomes. Certainly, treatment protocols have been based on genomic characteristics as seen here. Another application of high-throughput genomic technologies is in pharmacogenomics, to study pharmacologically relevant genes and to identify drug targets, as drug targets are commonly a protein that is intimately associated with a disease process. To wrap up, we have reviewed the biological background underpinning high-throughput genomic technologies and have seen some different hybridization assays. We have also looked at some interesting biological questions that we may want to explore. In the next several modules, we will assume that we have performed a microarray experiment and we will look at image analysis, alignment, normalization, expression quantification, and later we will cover topics in the analysis phase.